Hello.
Hey, how are you?
Good, how are you?
Good, thank you.
Um, so I don't know how to how do you review full lengths with students?
Um, were you able to do that blind review?
I tried. I'm not sure what
Hmm.
the Jack Weston like the
Oh,
Chrome extension you said, I don't have that. So I'm not sure if I'm doing it like the way that you wanted me to.
So wait, you're saying that you don't have the Chrome?
Yeah, I don't know what the Chrome extension is. Like I don't know if that's something because I never did a Jack Weston course.
Oh yeah, so it's yeah, so download it if you can, and it'll let us open the test with the solutions hidden.
Okay. Um,
Yeah.
because when I was going through it, I would just when if you don't scroll up all the way, scroll down all the way, you can see it. And so I was kind of just going through it and seeing if
Mhm.
I don't get if it was like a conceptual misunderstanding or just like a stupid mistake that that I was making while taking the test. And I realized like a lot of the times I'll give up on questions, like even if I understand the concept, like I'll be like, oh, I have no idea what this is, and then just like try to process of eliminate it. And
Mhm.
so sometimes it works, but like obviously most of the time it doesn't if you're like guessing on the test.
Got it. So I put a link in the chat for
Yeah,
the,
yeah, I know, I'm downloading it.
yeah. So yeah, once you
So, okay, all right. So, okay. Should I share my screen?
Mhm.
Okay. And are we sharing for YouTube today? Just curious.
Yeah, unless you don't want to.
Okay, no, that's fine. I don't care.
Um, yeah,
It's it's on the thing, right?
okay, so yeah, what you can do is review uh
I don't know. I was in ChemFiz when I was looking at it, but these are
mhm.
review all with Jack Weston.
Yeah, you could do that for any of the sections.
Um,
Yeah.
I don't know. I'm like really surprised that I got so many wrong on cars, but
I mean, it's likely just because you haven't been practicing cars for a very long time.
yeah. Okay.
I think the cars score is just because of that. Um,
Can we do like biochem stuff? Because I feel like ChemFiz I know is like one of my weaker points, but I didn't expect to do this bad on biochem.
yeah, I mean, the curve is different for the different sections. You can see like even a 1 point increase from the Chem Phys, but Chem Phys was 26 out of 59 and BB 32 out of 59. So you
Mhm.
got six more questions right just for that 1 point. So I
Still like a pretty weak score, I feel
well, you
like.
haven't taken it, this type of thing in a while, and you're just getting back to it. So
Okay.
yeah, I think it's fine. Obviously, we're gonna work on it, but yeah, I think it's fine.
Okay. So
Um, yeah,
which section do you want to do then?
whichever you want to do
I mean, I really want to like see what's going on here.
in bio, right? Okay, yeah, you could hit that review all with Jack Weston, and it'll give you like, yeah.
It's not running.
You might have to refresh because I think you just got that extension, so I think it needs like
Okay.
time to like activate or whatever.
Oh, okay.
So annoying.
Let me like not try for a second.
Yeah, yeah, what a bad time for that to expire.
Why does it do that?
I guess like people, there's always cybersecurity like risks, and various companies and stuff want you to set up two-factor authentication and also reset your password if it expires, but yeah, it's pretty annoying.
Okay. All right.
I just had a session with Mitchell right before you.
Oh, no way. He's still studying for it? I thought he was done with his MCAT.
Oh,
I lost the every time. How to get to the okay. I need to just
what part of the country do you live in? Oh,
I'm in New York.
oh yeah. What part?
Brooklyn, if you know where that is.
Yeah, I live in Bushwick.
Oh, no way.
Yeah,
I live down like by Coney Island.
nice.
Okay. All right. Here it is.
Yeah, okay. So this is yeah, so let's scroll down. So yeah, so which test was it? You can open it up, whichever test it was.
This one.
All right, yeah. Go to product. And then it should be like a big blue kind of button, but yeah,
Yeah.
you can click on it. Um, yeah, and
I
yeah.
know it works.
And see, so now yeah, you can hit show hide solutions. You have to put in your email, which is annoying, but that but yeah, you'll see what I mean when it this
Okay. This is the
so this is why I wanted you to do it. You see how
it's Chem Phys though. It didn't open the section that I wanted.
yeah, it's okay. But I just want you to see that this is the reason why.
Okay.
See how
I see what you mean. So it goes back to the like
yep. Because the way it is for without this is really annoying. Like it makes no sense that they do it that way.
okay.
But this way we get to see it exactly how it is when you take the test. Right.
So I learned about like microRNAs and biochem like a few weeks ago. So I was going off of what I knew from there. And then I honestly I can't remember what I picked as the answer when I was doing it if it's like this. So I feel like I'm not it's just like taking it from scratch.
Uh well, this is going to be kind of like what the purpose of like the blind review is, like to do it again, but with a blind review, you know which ones you got wrong. So if you want to have it show solution, you could have it do that. Um, we could also just have us review the ones you got incorrect. That could be a thing. So,
I know I flagged ones that I was like
uh-huh.
confused. Oh, you have to. Okay, how do you?
Um, yeah. So what you can do is in the filter on the left, you can filter by incorrect. And hit the yeah, hit the purple filter button.
I know. I know that I
Huh?
flagged certain ones.
That you what certain ones? Oh, flagged.
Yeah, like I flagged them, and then I guess three of them I got right, but
Yep.
okay. So I got this one wrong, but
Mhm.
do you want me to read through the passage?
Let's so is this something that you got um like I want to know if like you got like if this is the only question you got wrong for this passage or if there's a passage where you got a larger amount of questions wrong.
It
But
looks like I got the fourth one wrong too.
Okay. So yeah, we can go over this one.
So from I can't remember what I picked.
That's good though.
Okay.
Because like the blind review, that's like the purpose of that, right?
Okay.
So yeah, you could read out loud.
Okay.
And I'll be asking you about those buzzwords.
Okay.
Okay.
MicroRNAs are short, non-coding single-stranded RNAs, approximately 19 to 25 nucleotides in length that bind to mRNAs. Numerous protein encoding genes are regulated by microRNAs, including those involved in the immune response. Research has shown that rejection of transplanted organs is associated with alterations in the expression of various genes. To determine whether a link exists between microRNA expression patterns and organ rejection, a team of scientists performed the following sets of experiments.
Okay, so you're reading that really fast. Can you tell me what they're kind of trying to say here?
And we were talking about mRNAs, sorry, microRNAs, and then they were talking about organ transplants and like whether there's an association between microRNA expression and rejection.
Yeah, so okay, we can write these relationships out. Actually, let's see if you could zoom out a little bit more. Oh yeah, that's perfect. So, okay, so these microRNAs are these small RNAs, I guess, can do that can affect uh what um in this paragraph.
Sorry, what?
So so do you remember when we talked about proportionalities?
Yeah. So,
So so what I would write here would be, so we know that, so it says research has shown that rejection of transplanted organs is associated with alteration in the expression of various genes. Um, and prior to that, they said that microRNAs bind to mRNAs, and that can affect what genes get expressed,
mhm.
right? So, um, so I would maybe just like think about like, just how this can work to like stop the expression of something, right?
Okay.
But yeah, you can continue.
MicroRNA levels were measured in the biopsy specimens of patients with normal transplanted kidneys
Oh,
and patients with.
we're up to, oh, sorry. You did read it, but I forgot. So just over here, we're saying that um like we're essentially saying like we're going to do an experiment to see if there is a link between microRNA and organ rejection, right? And so that's why they're doing the experiment.
Okay.
Yeah.
All right.
Mhm.
Um, and patients, or I'll just read it from the beginning. MicroRNA levels were measured in the biopsy specimens of patients with normal transplanted kidneys and patients with kidneys that showed histologic features of acute rejection. MicroRNA sequences are shown in Table 1.
Okay. So on the test, spend like 15 seconds looking at this table and see if you can find any type of, meaning in it. So it looks like we have various types of microRNA and the various sequences for the microRNAs. And I don't really think we
I
can.
think this was for a different one of the questions, but it was one that was asking about like the strength of the bonds. And I know that like
Yeah.
CG is like a tertiary, like has the three, and then A to T only has, or I guess A to U, okay, anyways,
Yeah.
only has two.
Yeah.
And so I it definitely wasn't for this question, but I like drew it out and I was like, okay, so there's like this many here and this many here, and so this is what it means for the. I guess I got that question right because
Yeah. So on our first, pass through, we don't want to spend too much time on it. So
yeah.
like 15 seconds, like, okay, we have a type of microRNA and the sequence for it. And there's nothing else we can really figure out from here. But if they ask us about it, we can consult the table, right?
Okay.
Um, and yeah, okay. And then the next one.
The results for the microRNAs analyzed are the results for the microRNAs analyzed are shown in the figure one. Expression levels of microRNAs in biopsy specimens of normal and AR kidney transplants. Of these mRNAs, these ones are highly expressed in normal lymphocytes and monocytes, whereas MI.
So tell me a bit about this figure here. Um,
So
you could
it's like showing the normal transplant and acute rejection, and then like the scale of like the normalized expression. And so it's a lot lower, or I guess it varies, but in
Yes.
certain ones, the normal transplant ones are higher, and in certain ones, the acute rejection, they're higher.
Yeah, so just scroll up a little bit. I just want to see like what they said right before. Okay, so just the result. So if we look at this graph here, or chart or whatever, I always think it's best to, I mean, obviously, again, if this is on the passage, 15 seconds, but sorry, if this was on the test, 15 seconds, but since we have a little bit more time here, we can actually look at this and see if we find any type of pattern. And I think you were saying something like along the lines of
Which it says like right here that the first three
Oh, okay.
normal lymphocytes and monocytes,
Yeah.
and that's associated with like the normal transplant, and then highly expressed in normal renal cells.
Mhm. Yeah. So like yeah. So before that part, you could see in the chart how yeah these first three microRNAs um like uh
They have an association.
yeah with AR, which is acute rejection, and then the other three microRNAs um will be in normal transplant. So okay, good. And when they say normalized, what do they mean exactly?
I mean that they're not like mutated or changed in any way. Like this is just what the mRNA levels normally are.
So it'll just be something that's used as a standard that you equate to one. And so everything after that will be like a percentage or a fraction of that one. So they're saying that in the caption that microRNA levels are normalized to the small nuclear RNA RNU44. So that means whatever this value is, like let's say it's like 80, right? This normalized levels would be like for instance, like this number one here would be 80, right? This, over here would be like a fifth of that, etc. So normalized, you'll see it a lot in graphs and charts, and it just means you're taking something and you're equating it to 1 or 100, and everything else is a factor of that.
Okay, that makes sense, especially for like experiments and stuff.
Yeah, yeah. So, okay, good. And then you read that these are highly expressed in normal lymphocytes and monocytes, whereas the those other ones are in renal cells. Oh, okay. That's interesting. So, uh yeah, renal um lymphocytes, monocytes, okay?
Okay. Using AR biopsy specimens, researchers measured the levels of CD3 mRNA and CD20 mRNA and determined that there was a positive correlation between these mRNA levels and the levels of the microRNAs, but not those microRNAs. Researchers evaluated the glomerular glomerular function of the patients.
So sorry, so let's talk a little bit about experiment two here. So what's going on here?
They were measuring this T cell and B cell mRNAs and looking for like a correlation between them and the microRNAs.
Okay, so and what did they find?
They found a positive correlation, but
Yeah.
uh between three of them.
Uh-huh. So basically um the so this one would be the first three as well, right?
Mhm.
So and positive correlation is they usually use that like in the sciences they usually use proportional or inversely proportional, and in
Mhm.
soc they usually use like positive correlation, negative correlation.
I've seen like double reciprocal a lot recently.
Yeah, yeah, yeah. There's when you say double reciprocal, do you mean
Like it'll be like
like a
I've seen it in a question. It was the double reciprocal, the this and this are in a double reciprocal plot correlation with each other.
Lineweaver-Burke?
It must have been, but I don't think it like explicitly said that it was Lineweaver-Burke in the question.
Is it like something like this or something?
Well, that is like Lineweaver-Burke, but I don't remember the context of the question. I just remember that it said like double reciprocal.
Got it, got it. Yeah, so positive correlation is basically telling us that it's a direct proportionality. So if there's so I would write it as proportionality. So that's what I like to do all the time anyway. So I would say that let's see, T slash B cell um mRNA proportional to um micro RNA like 142, 155, 223. Right? All right. Um, yeah, and then the next experiment.
Evaluated the glomerular function of patients at the time the biopsies were taken and observed a reduction in kidney functions for patients with AR transplants. So AR was
Acute reject.
acute rejection, which makes sense.
So tell me what they're saying in experiment three here.
So I'm not sure what this word means,
Yeah.
but I'm assuming it has something to do with like output or like kidney function, just connecting context clues. But it also makes a lot of sense that kidney function would be reduced in patients with like failure, acute, I forgot, I keep forgetting what the word, like acute rejection. Like it makes sense that if the body's rejecting the organ, that like the function when even if the liver isn't or kidney isn't the organ that's being transplanted, like it takes energy away from the other organs to.
So you can maybe say something like that.
Mm-hmm.
Acute rejection means that you're having less of a less kidney function, right?
Okay.
Does that make sense?
Yeah.
Okay, so um, since you mentioned kidneys before, I can tell you what the glomula, however you pronounce it, is. It's like the bundle of capillaries that isn't that the Bowman's capsule goes over, and that's where the filtration happens from the blood into the start of the kidney tubule.
Okay.
Yep. So,
Sounds
okay,
good.
any questions so far here?
I guess not there, but my questions come up when you look at the answer choices because for me this one was like a guess and I was trying to like from what I remember make like an educated guess by ruling stuff out, but as far as like defining each of these answer choices, I'm not sure what like adaptive or
So let's think about
like because it has I let's we can read the question, but like because it has to do with like rejection, I assume that it meant like adaptive because something was changing in the body.
so what yeah, so what it from the passage, what types of immune cells do we see?
T cells and B cells,
Anything else?
I,
Mhm.
like lymph nodes and monocytes.
Oh, lymphocytes and monocytes?
Yeah.
Yeah. So I think that's what they're trying to do here, like confuse you about whether it's lymphocytes and monocytes or T cells or B cells. Um, but they said that the lymphocytes and monocytes are for the
It's only for 3 of the microRNAs.
mhm. Yeah. So this is going to be, oh, and then were you saying like that you didn't know some of these or
Yeah, like
Mhm.
I'm not sure like these immune responses. Like I can take like guesses as to what they would be like.
Uh-huh.
Adaptive would like mean a change in the body, and so innate is just like how it normally functions.
Uh-huh.
It's the innate response to things. So I'm almost positive the answer choice I picked was D, just from like an educated guess.
So let's talk. Okay, what's the innate?
So is innate just not just like the normal function of like the immune response?
What?
That's just what its job is.
Tell me what you mean when you say the normal.
Like if the body identifies like a virus or something, like these immune responses get triggered to go against them. Like that's the innate response. Unless I'm like completely off here.
No, no, no. You're right. So innate would be which of the types of white blood cells that they showed us here.
I believe T cells.
So if innate are the ones that you're born with, um, think about that like a little bit. Like, so like normal lymphocytes and monocytes, those are from the innate.
Okay.
Yeah. Because the innate system is the one that everyone essentially has, and it's like um well, there's like
But
neutrophil.
we always have white blood cells in our body, no? Like,
That's I kind of foresaw that question coming. Well,
or there's like they multiply when they're needed.
well, it's a specific type. So like if I were to use like an analogy, right? Um, the phagocytes in the innate immune system, like neutrophils, macrophages, basophils, eosinophils, and natural killer cells, those are like the first responders. Those are like there's an emergency, something happened, we have these bad guys coming in, whatever. And so you have like EMT workers and police, firemen and stuff, and they're first responders. Like they're gonna be there first, they're gonna attempt to deal with it. And they're able to be mobilized very quickly,
Mhm.
right? But acquired, also known as adaptive immunity, is when you build a resistance to it, which means that you get infected and your T cells and your B cells, like, so I knew you were going to ask, like, isn't that always there too? Uh, to an extent, yeah, but the T comes from like the thymus. I believe it's a thymus where they mature. So all the blood cells originate from the bone marrow, right?
Mhm.
But but some of the white blood cells can move elsewhere to be matured. And so like for the T cell, I believe it's a thymus. I could look into that. The B cell, all these things are going to be specific to that pathogen.
Okay.
So
So if we're operating off of the assumption that like these like normal lymphocytes and monocytes already there, like this isn't part of innate, so that immediately rules out C and D, right? And then adaptive, I still think is correct because it's like something is changing in the body. And
Yeah, because we're dealing with T cells and B cells, those are what adaptive immunity is basically.
uh-huh. And what is humoral?
Humoral is it's like there's cell mediated and then there's humoral. Let me make sure here.
Is it like a just in case kind of a thing? But that would be more innate.
So,
So cell mediated and humoral, those are both answer choices.
yeah. So,
Well, cell mediated would be more specific, I'm assuming. Like that's like on a more like cell to cell basis. Like if a cell's being attacked, then it'll send out like some response.
yeah, I think that the humoral, one sec.
So I guess the answer choice that I picked was like a really silly one because like by definition, this would like they would cancel each other out.
Well, you could have, I mean, if we had normal lymphocytes and stuff in
They
a,
would still be there to target it, but like they were saying that they're like highly expressed in like those normal mind like RNAs.
Mhm. Yeah. So okay, cell mediated is going to be uh involved with T cells, all the different types of T cells, so helper T cells, cytotoxic T cells, memory T cells, and what they do is kill bacteria or kill a pathogen that's already invaded a cell, a host cell.
Okay.
So cell mediated, think about, okay, it's the virus or the pathogen is within the host cell, so we need to deal with
Hmm.
that host cell. Whereas humoral is dealing with an extracellular pathogen, and that would mostly involve I mean, they'll both involve B and T cells because they work together, but but yeah, cell mediated would be for a pathogen within a cell, and humoral would be, you know, outside of the cell.
If there was an answer choice that was like adaptive and cell mediated, wouldn't theoretically that also like they could go hand in hand and like would you still pick only adaptive in that scenario is I guess my question.
So, well, we know that sorry, so you said if it was
Because
uh
I feel like from what you're explaining now, it seems like they go pretty hand in hand, like a cell-mediated response. Well, also humoral too. Like, how do you, I guess without knowing the specifics of it, like adaptive is the best answer choice, but like I just I don't see them like canceling each other, like negating each other.
yeah, so okay. So what when you gave me the that choice that you came up with, can you tell me that again?
So I was at first I said cell-mediated and adaptive, but also humoral would I guess work with that too.
Yeah, so okay. So yes, adaptive includes humoral
All of the other
and
one.
cell mediated, because otherwise, why wouldn't it be B, right?
Hmm.
But that only is what's wrong about B, because what we have is humoral plus cell mediated, which is the adaptive. C says cell mediated, which we can certainly have, but innate is not the one that happens in this experiment. And
Hmm.
D, adaptive and innate only, that's kind of everything. Like you can't have something that's not adaptive and not innate. So that doesn't really, help us out. So yeah, it ends up being adaptive. But if you were to change some of these words in the choices, then that's could be a good exercise as well. Like if I changed innate here and wrote humoral for C,
Hmm.
it should be right.
But
Yeah.
wouldn't that be the same answer choice then it does A?
Oh,
Because
I'm not really like, I'm just saying like,
oh,
if,
like you're saying that like that would be okay, I understand.
yeah,
Okay.
yeah. So sometimes I like to do that because it makes you could get more bang for your buck for each question if I change like little
Mhm.
things.
Yeah.
It could also be useful for you to be like, okay, this is wrong specifically because of this word.
Hmm.
Stuff like that. Yeah.
Okay. Um, this was a stupid question I think to get wrong.
Well, tell me about it.
Um,
Hmm?
I can't remember what I picked. I think I said C.
Mm-hmm.
I just, again, I feel like almost my class stuff has been messing me up a little bit because now I'm like, oh, my professor would say that like all like natural life is like regulated by H bonds.
Well, H bonds play a major. I mean, that's why water is so important. I mean, it's important for a lot of reasons. Don't get me started on that. But there's hydrogen bonds come up all the time. Um, but they
Is
are
the phosphodiester?
so. Before I say anything about that, hydrogen bonds are intermolecular bonds, right?
Mm-hmm.
And would you draw them as like a line or like maybe a dashed line or something?
A line.
So I mean, like, let's say we have this and this. There should
Mm-hmm.
be a hydrogen bond between this oxygen and this hydrogen, right?
Mm-hmm.
So would I draw that like this, a line like that? Or would I draw maybe like something like that?
Well, isn't that like a, I can't even remember what it was called. Like when the when you had like the order of like it wasn't electronegativity. I don't know what I'm thinking of right now, but how like the hydrogen would be the fourth resonance, right?
Okay.
That's what I'm thinking of.
So,
How would resonance like that isn't the wedge and dash line based off of that?
mm, not really. The wedge and dash is just to show you, like, is to try to show you a molecule in 3D with
Mm-hmm.
wedge being like into the page and out of the page. But okay, so like if we think about a regular like ionic bond like NaCl,
Mm-hmm.
that would be Na bound, oops, to Cl with a line, right?
Yeah.
Now let's say we have um H2O, right? So that would just be, sorry, not running out of room, but oxygen and two lines to H's, right?
Mm-hmm.
And that's a covalent bond, right? However, oh, and these are intramolecular, but if you are figuring out what type of bond exists between two different water molecules, it's not, if it was a line like this, that's not going to work, right? Because it's not going to be able to make an additional bond with hydrogen. So it just makes a hydrogen bond, which is an intermolecular type of force, and it's not as strong as actual ionic or covalent bonds. I mean, it's the strongest type of intermolecular bond, like meaning you're gonna have um like there's like four types of intermolecular bonds. Hydrogen bonds are the strongest, and that's when you have hydrogen along with something like FON bond.
Mm-hmm.
So those are going to be the most strongest. There will be dipole interactions, which could also be considered like forgot the name exactly, but it's like a electron, like a yeah, I forgot the name exactly, but it's based there. There's that, which is essentially what we have with NaCl here. Um, so and then you can have van der Waals, which is also known as London dispersion, which is the weakest type, and that just is based on instantaneous dipoles that could exist.
Yeah.
So basically, separate the intermolecular bonds from the actual bonds. And actual bonds, which means covalent or ionic, we draw as lines. But if we were to draw hydrogen bonds or any of the intermolecular bonds, we can't use a line because it's not, because a covalent bond, right, would be like both of the atoms sharing that electron or sharing an electron, right? And ionic bonds would be only one of the things that's kind of hogging the electrons, but intermolecular, you're not changing, you're not doing a bond, so you're not changing like the number of electrons of something, basically.
Okay.
So, um, so okay. So hydrogen bonds here are the only ones, is the only question that, or choice that has an intermolecular bond, right?
Yeah.
Now A, B, and D, these are all straight up covalent bonds that you would draw as with a line, right?
Okay.
So yeah, so we have disulfide bridges. What do those look like?
I can visualize it. I can't explain.
What do you visualize?
Like on when I'm looking at it's like two. Ugh, I don't know. I don't know how to explain.
Okay. So let's say you have two cysteines with SH bonds, aka thiol bonds, and you oxidize it, you get your disulfide bond.
Okay. And so
Yep.
like it's wrong here because of like the structure of them.
So before that, I just want you to see, so this disulfide bond, is it an intermolecular bond or is it a
No, it's covalent.
good?
It's between the two sulfurs, right?
Yes, yes. And by the way, if you wanted to go back to this, you reduce it.
Mm-hmm.
So that's what happens in reducing like gel electrophoresis,
Mm-hmm.
break the disulfide bonds. So okay, there's that. And then glycosidic bonds will be the bonds that connect,
Carbs, right?
yeah, sugars, and they will be like ketals or acetals,
Mm-hmm.
um, which are not reducing, because I like to ask if something's reducing sugar or not. And yes, the last one is phosphodiester, and
That's
that,
links the ribose sugars.
yeah, right?
So
Because
that makes sense. That's like anything with like DNA and RNA is phosphodiester linkages. Okay.
yeah,
If I like mark it correct, does it say like
it'll show you the solution, I think. If you hit the button. Yeah. So I'll erase
oh,
it.
but it doesn't say like what I got.
Your previous answer is C.
Was it? Oh,
Mm-hmm.
yeah, so exactly.
Yep, yep. So yeah. So you can see how this extension is good for this as well as like the explanations
Yeah.
are a lot better.
I see that they're like a lot more in depth than
Yeah, because the AMC explanations are the worst ever. They're just like A is right because A is right. And they're just like,
and
okay.
then they have like a little Khan Academy video if you like need to
Yeah,
understand the
yeah.
topic. I hate it because it's like these are the skills that you're wrong on.
Yep, yep.
Okay, should I read the passage?
Oh,
Is
so
there
how many did you get wrong for this passage? Two,
two,
three.
three?
All right, let's do that one.
So are we only doing the ones that I have like a lot of questions wrong on?
So that's, I mean, we could do anything you want, like at all. But the reason I'm doing this is like, if we spend the time to go over a passage and you only
Mhm.
got like one question wrong, it's like,
It's a little bit of voice. I understand. Okay, let me read through it.
but yeah, if you have any question that you have or specific passage or anything, you just let me know.
Okay,
Yep.
sounds good. The microbiome has been investigated for a possible link to non-hereditary diseases. Crohn's disease is a non-hereditary chronic inflammatory condition that may affect part of the GI tract. Causes for CD have not been defined to date. However, research has shown that the profile of the GI tract microbiome is different in healthy versus CD affected individuals. So, like gut microbiome, I learned a lot about this year. Like I was taking a like nutritional science class. So, um, therefore, researchers hypothesized that differences in bacterial distribution and the host immune response to GI tract. So, bacterial distribution and host immune response. Bacteria might play a role in the establishment of a progression of the disease.
Okay. So, like many first paragraphs of science passages, it gives you like some generalized clinical background. But it doesn't tell us anything yet that we can put into proportionalities because, like, they're saying that it's like, you know, differences can play a role in the establishment and progression, but we don't know if it's like going to make it go up or down. We just know that it's being affected. Like, there's a relationship between CD and bacteria in GI.
Okay. So we're like looking, so this is giving us the background, but not like the method.
Uh, yeah, just like a general, like just to be like, hey, this is still, for people who want to go to medical school.
Mhm.
Because otherwise you're just going to go right into it, and people are going to be like, why do I need to know this? Um, but good. Okay, yeah, you can continue and yeah.
GI tract bacteria digest dietary fibers and convert them to butyrate, propionate, and acetate.
Okay. Now we can do something with this, right? So we can say what how would you maybe write this or express it?
These are enzymes that like regulate certain acids, right? So this is probably some like digestive enzymes.
Uh well, what is it saying that it is?
Dietary fibers and oh, they convert them to
Yeah, what does?
the GI tract bacteria.
Okay, okay. Because you were saying enzyme, so bacteria, right? So we can say something like fiber through the action of bacteria can give you like butyrate, propionate, acetate. I'll make
Okay. Should I keep reading?
um oh yeah.
Okay. These three molecules modulate the innate immune system response by attenuating the inflammatory response to GI tract commensal bacteria.
Um so these three molecules, modulate the innate immune system, okay, by attenuating the inflammatory response to GI tract commensal bacteria. What are they saying here?
Like, so they like make sure that everything is working properly in the immune system response. And then if there's a certain like, I guess, bacteria that like causes a lot of inflammation, they'll like handle it.
Yeah. So I would maybe say like inversely proportional to.
Inflammation.
Mhm.
Okay.
Good.
Furthermore, butyrate is a major source of energy for colonocytes, which is probably like some sort of bacteria in
Yeah,
the
they'll
gut microbiome.
probably ask something about that, but yeah.
The GI tract microbiome of healthy individuals exhibits a predominance of gram-positive bacteria and a reduced amount of these two, which probably gram negative. Or no, they're all gram positive.
Mhm.
Okay. Uh, oh no, they are gram negative. It says right here. This is in contrast to the microbiome of individuals with CD who have an increase in the number of gram negative proteobacteria, whereas the number of firmicutes bacteria is highly reduced.
All right, let's take a look at this. So okay. Um,
So healthy has a positive association with gram positive,
mhm.
and then CD positive association with gram negative.
Okay.
Okay. And then treatment with 5-aminosalicylic acid reduced proteobacteria without affecting the number of other bacteria in CD affected individuals. So that's like they're treating the gram negative, like the increase in gram negative with the 5-aminosalicylic acid,
Mm-hmm. Proteobacteria.
which is gram negative.
Oh crap, yeah.
Yeah. Well,
Yeah.
never mind, yeah. Okay. Um, Bacterial characteristics and the variations in microbiome between healthy and CD individuals are summarized in table 1. And so then it's listing out all the, like the negative, and then it has the energy sources and the products.
All right. And so bacteria analyzed in the large intestine, we have the different ones. We have if it's positive or negative, um, energy source, product, and the asterisks show you the different. Oh, they're a little different. Usually it's like p-value less than 0.05, but here it's telling us that a single asterisk is highly reduced. I guess it means highly reduced like proteobacteria maybe. And the double asterisk indicates highly increased number of bacteria in CD individuals compared to healthy. Okay, and then so PS indicates polysaccharides, LPS lipopolysaccharides. Okay, there's a lot of stuff there. So, but we did our best. We get to see what they're trying to tell us here. So yeah, we can attack the uh question. I will be right back. I'm just gonna use the bathroom. Sorry. Okay. So, all right, let's try this out. Yep.
In which metabolic processes are the GI tract bacteria directly involved? Uh, Honestly, I don't remember what I picked for this one.
It's okay. It's better to forget, to be honest, because you got a sort of a fair shot at it. But um,
Uh-huh.
and also when you do your blind reviews, I want you to do that also, like without knowing like what you did before. But yeah, let's try this out, right? So
Mm-hmm.
um, so yeah, how would you go about this?
I guess I would look at
Hmm.
the table and look at the energy source and the outputs and kind of see what information we're able to draw from that to answer the question. I don't know. That seems like so it's taking like acetate and whatever else. And then so I feel like immediately looking at it, A is wrong because that's only the energy source for like three of them. So I don't know. I feel like that's probably, I don't know. I guess we'll just table it for a second because it does come up a few times. Um, I don't think it has anything to do with like the absorption of amino acids because the only time amino acids came up in the passage was the 5 amino salicylic acid. I don't know.
Okay.
I guess I don't know. PS is polysaccharides, right?
Mhm.
Okay. So then, well, I don't think D is right because I feel like monosaccharides aren't usually absorbed. They're like polysaccharides are broken down into monosaccharides. Oh, for them to be absorbed. I don't think that dietary fibers are usually fermented into peptides though.
Mhm.
I honestly like it would be a total like into the dark because I think I'm gonna change my answer and go like the fact that it does appear to be like an energy source in a few of them and then they're like broken down. I don't know, these just all kind of don't add up.
Okay, let's see.
It could also be D. I don't know. Like I
So table one, let's try to think, okay, what is table one trying to show us, right? So we know all of so it says bacteria analyzed in the large intestine. So we know all these bacteria like count. So we need to look at them. And whether they're positive or negative doesn't matter for this question. But then when we look at energy source and product, that is what will help us. So let's see. So first one, we have acetate turn into butyrate. Second one, succinate turn into propionate. Oh, and we know from the passage that where if you break down fiber uh, by using the bacteria, that you'll turn it into butyrate, propionate, or acetate. And we see that for the product for the first two. Um, the third one has PS, polysaccharides, as an energy source, and it still produces acetate. Um, and then we have glucose and its isomers, acetate, propionate, butyrate. But we also have some stuff like H2 gas, hydrogen sulfide, which they didn't really, yeah, that's not something that they're really that they listed as some type of product like in the passage. And then we have PS plus glucosamine gives you butyrate, glucose, acetate, PS lipopolysaccharide, and lactate. They didn't give us that either. So they didn't give us that, or that.
Mhm.
So those are not going to be what we're concerned with, but we want to find something that takes this and turns it that, into that product, right? So if we look at um, absorption of monosaccharides.
I don't know if that's specifically the bacteria is wrong.
Yeah, because the bacteria here is breaking or converting energy source to product, right?
Mhm.
So the absorption of the monosaccharide, that means the monosaccharide has already been broken. Like you start with a polysaccharide, you break it up into monosaccharides, and then the monosaccharides can get absorbed. So D won't be it because the monosaccharide indicates that it's already broken down, and absorption is what you do after you break it down. But we're looking at bacteria that actually do the thing that breaks it down,
Mhm.
right? So D,
I feel like I don't know. I guess A is the best choice here, right?
so let's let's see. So
You want to go through them one by one?
yeah, because I want you to be like to get to be able to get questions without relying too much on luck. Like it's more than just eliminating wrong answers, it's eliminating them for the right reasons.
Mhm.
So like for C, fermentation of fiber into peptides, they mentioned fiber, right? But they mentioned fiber being turned into butyrate, propionate, and acetate, which are not peptides. So that won't work. And then absorption of amino acids is wrong for the same reason D is wrong, because the bacteria isn't causing absorption, it's causing the breakdown of things.
Mhm.
So A is conversion of polysaccharides into short chain fatty acids, and I guess it might be something that isn't um like universally known, but there's like a passage in the section bank that's similar to this, and it basically states that these are the short chain fatty acids. The butyrate, the propionate, and the acetate. This is basically like what happens with ketosis, like
Mhm.
a ketogenic diet or something. So yeah, that's what we see. So basically for this question, what we did was we are looking for some type of chemical reaction. So B and D, the chemical reaction's already happened and it's just causing absorption, so those are out. And then C is close with the fiber because the passage mentioned fiber, but it doesn't turn them into peptides. And so A would be it. And if you didn't know what that short chain fatty acids were, these things, it would still be better than the other choices because we know that it's not going to be peptide that is produced. So yeah.
Okay. I'm curious.
Mhm.
Is this week? Yeah. I picked C.
Yeah.
So yeah.
I was gonna predict that to be like because they use the word fiber.
Mhm.
So a lot of these types of things, like you can get to the point where you can predict the trap answer choice. So this time it was this, because literally the passage used the word fiber, right?
Yeah.
So that's why I would imagine a high
But
percent.
I feel like the fact that it said based off of the table and not the passage should potentially help.
It could do that. It could do that. Um, but
But all right, I now I understand why that's wrong because it doesn't turn them into peptides.
mhm.
Okay. Based on what is most likely the association associated with a reduction in gram-positive bacteria. Um, we said that gram-positive was associated with the healthy, so a decrease in gram-positive would mean that there's like not as like well, okay. Just like these ones would go up, these two, proteobacteria. So then I could look at the table and see maybe what factors are associated with.
Mhm.
So the double stars is so we would be looking at the two-starred ones, right? Because those are the ones that are higher in CD affected patients.
Well, what we really just want to find out is
Oh, but we want the gram negative. So I'm gonna say that this decrease in PS production is wrong because polysaccharides are obviously still a source of energy in the gram negative ones.
are you looking for gram negative or gram positive? Like,
Well, it says association with reduction in gram positive bacteria. So I wanted to see what's like associated with gram negative.
okay, well, we don't know yet if
Well, okay, even if we're gonna look at the gram positive, there's I guess my point is that it's associated with both like for the polysaccharides, it's associated with both the positive and negative, so I don't think it has anything to do with like its production.
okay.
I
So
don't think it would necessarily decrease.
okay. So let's see. So if we want to look for something that causes a reduction in gram positive bacteria, so yeah, so I would so that and that. And what we have in this is basically energy source turning into product.
Yeah.
So the first one is acetate turning into butyrate. Doesn't really help us. Second one is polysaccharide plus what?
Hydrogen.
Okay. Um, when you say hydrogen, you mean
It says H plus, no?
like a proton? Like that's what you mean?
Isn't it?
Because yeah, because you just said hydrogen, which would be just hydrogen, but this means it's an acid, isn't it?
Mm-hmm.
Right? And if something is going from like this to this,
Mm-hmm.
right, it's gonna reduce the thing that it kind of, starts off with, right?
Right.
So, and if we look at the next one, it's just glucose turning into acetate. So, yeah, so we want to see what would cause.
So wait, you just said something that like kind of answers it no, because if it's a reduction from this to this, then a reduction of a gram positive bacteria would create one of these answer choices. So if you look at which one comes out,
Mhm.
is it well, okay, A and B are like I still think D is wrong. I don't think it says anything about pH, but between production and use of, energy source would be using and production would be. So is it A?
Acetate production. Well,
Oh, wait. Well, it is here it is the product,
uh-huh.
but it's also being used, which I think is a trick that they're both wrong.
Yeah,
I
yeah,
guess C maybe because that's like the least, I don't know, for me, like it was the one that drew my eye the least. I still don't think D is right because decrease in production. Oh, but it isn't being produced anywhere.
uh, it, I guess it'll be produced by just something that's not here. I think that's what it might be trying to say.
But how can you tell that based off of the passage? Like where does it like let you assume that? Like
Oh,
if you're looking at it as an energy source, then it's present
mhm.
across. But it's
Yeah.
like
So,
in my head when I was thinking of like reduction, like it wasn't like a redox reaction. I was thinking like if they took away like this amount of like the gram positive bacteria, which would mean that there would be more like gram negative, like what effects would it have on like the overall product?
uh-huh.
And so that's why I was like, if there's more negative or positive, it's still using the polysaccharide. So I don't really think.
So,
But as far as a product, it's not a product anywhere.
so when they say reduction, it can be confusing, but it can be really confusing about, do they mean reduction in redox or do they just mean less?
Mhm.
Which definition do you think they're using here?
Immediately I went to less of, but now I'm
Yeah,
thinking
that's right. That's
redox.
right. That's right.
Oh.
Yeah. It's annoying when they do that, but they just mean, yeah, what's likely to happen if you end up with less uh gram positive bacteria.
You wouldn't be able to produce as much of the product.
So let's see, right? So A, increase in acetate production. We do see that here, right?
Uh-huh.
Um, And
But that's, if you're getting rid of it, then there would be less of it,
yeah, yeah.
of acetate. So it wouldn't be an increase, it would be a decrease if you got rid of it.
Yep, yep. So okay, so that would be the acetate production part, and this would be the use of
Of
acetate,
acetate.
and kind of like what you said, like they're both like
They
saying
negate each other because they're both right.
yeah, yeah,
Well, they're both wrong.
yep. So now remember when I asked you about the other one? Like I asked you a little bit more about it?
About P C or D?
About oh, just that one?
Sorry, I have no idea what you circled.
Oh, sorry.
Oh, okay. Yeah.
Tell me your thoughts.
Oh, because you were asking about like,
Mhm.
are you just like, I don't I guess you're like pointing to it being D, but I understand that C doesn't come up anywhere in the passage, but is it something that like you can deduce like based off of certain factors that are here? Like if it's more like gram positive or gram negative that says certain like things about like the overall pH.
Right here,
Yeah,
right there.
I see. Yeah. So if you take it out, then but isn't that like charge associated, not
If there's so that means that it's acidic,
mm-hmm.
right? Right?
Right, but that's a positive one. So if you're using it to acetate, you're making it more acidic by using the hydrogen.
Yeah, so if you had so this gram positive back to like rumen whatever, in order for it to function, it will consume um polysaccharides um as well as the protons that are given away from an acid, right?
Mm-hmm.
So if you were reducing the amount of this rumino bacteria, you're not going to have that energy source be used up as much. So you're going to have higher levels of H plus, which means more acidic, which means lower pH.
Which is an answer choice.
So what I want you to see from this is that yes, they may not have spoken about or mentioned pH, but that doesn't mean that we can't infer something, right? And so that's why when I asked you about this over here, I was kind of like spending a little bit more, time on it just to be like, all right, what does that mean, But but yeah, a lot of times that's why like I like it when students would use their own words to describe stuff, like particularly in cars, but like in general, that just helps you be able to see that because if you were just looking for the word pH, yeah, you're not going to find it. But if we have an H plus here, then pH is involved. Yeah.
Understood.
Mhm.
Okay. Which also makes sense with like the stomach, how like
Yeah.
or
Yeah. So yeah, yeah.
all right. I'm just curious as to what I picked.
Mhm.
My answer was A,
Mhm.
which is wrong. Okay. I was looking at production. I saw acetate a few times, so I was like, sounds good.
Yeah.
Microbiome of CD affected individuals. So now it's CD affected is the starred ones. So these two. Acetate and lactate. Um, I think C's wrong. Doesn't say anything about propionate. Um, immune tolerance isn't necessarily a factor, so I'm gonna like put a question mark on D for now. Increased digestion, potentially, but not necessarily any different from like normal.
Yes.
So not sure about A. With B, dietary fibers and convert them to. I've been defined that the profile is different.
Mhm.
So it doesn't say anything specifically about like di I want to say I picked B,
Okay.
but that still seems correct to me. Like I know it's not necessarily slower, but just like using context clues.
Uh-huh.
You would imagine that somebody with like Crohn's disease that wouldn't be able to absorb fiber as well. But I don't know, are you, if it's a passage-based science question, are you allowed to use like other information that you have?
What, like,
Like,
you're
you know how in cars, if like you know something about the topic, but it's not like explicitly said in the text, it's better not to like use it to answer the questions.
so cars is the epitome of don't bring your own knowledge into things. Sometimes it can help to have a vocabulary to digest some parts of the passage, but you don't want to bring in your own knowledge of whatever topic it is. But for the sciences, of course, you're going to bring in your own knowledge, but it also has to be like valid in the passage as well, right? So it's a combination of both for the sciences. You need to know the stuff and you need to process what they're telling you in the passage and all the data charts and stuff and apply what you know to those to that type of situation to the whatever question that they're asking you about. And there's lots of um yeah, since you're good at cards, you're going to be able to notice really small nuances in language. Like for this one, there's like, okay, so you mentioned B. From the previous question, do you remember me eliminating like two answer choices because of a word, a particular word?
There was something to do with like, was it these two that we were eliminating?
Oh,
Or
sorry,
the one
I guess
before?
for that.
The one before that where it was like
Yeah,
monosaccharides and amino acids or no, you did.
yeah,
No, it was B and D that were like not related specifically to the bacteria's function.
yeah. Why?
Um, that's I can't remember specifically what they do. It's something with like that's
So,
like it happens in the like gut, but it might not be regulated by bacteria, like from the information we have.
so basically, let's say you eat some ice cream or whatever, you get uh carbohydrates in your body will break down the carbohydrates into
Mhm.
monosaccharides, things like that. And that's separate from absorbing it, right?
Okay.
So
Yeah.
in this passage, the bacteria is actually digesting and breaking apart the whatever it is, but
Hmm.
that's different from absorption. Absorption is after it's already been broken down.
Okay.
That's why for this choice, I mean, for this question, I eliminated B and D.
Because of the absorption word.
Yeah. And because they're asking what do the bacteria do?
Hmm.
So yeah, if we go back to the next question here. So what would I not like about like one of these choices? So it's the same word as the previous one.
Well, the one before you didn't like the word fiber.
Oh well, was it that or was it a different word?
Absorption,
Yes.
the two before absorption was your issue.
Yes.
So we're that, okay.
Because absorption is like the simplest form of those for proteins it's amino acid, for carbohydrates it's monosaccharides, fatty acids for triglycerides and stuff.
Mm-hmm.
Absorption is the absorption of that from your GI tract through your villi and entering into your um, bloodstream, right?
Mm-hmm.
So that's the absorption part. So but all of the stuff that we're seeing that the bacteria do are like legit chemical reactions. Um so B shouldn't be an answer because of the word absorption, because that's not related to what the bacteria is doing.
Mm-hmm.
But
But
it
it's in this one, it's not saying anything about the bacteria. But I guess, oh, the microbiome, never mind.
yeah, and just like I say, like it's good to have legit reasons for eliminating wrong answer choices. If you can even have more than one reason, that's even better. So dietary fiber, I didn't mention anything about it before, but fiber, we're told in the passage, is broken down into this other stuff.
Mm-hmm.
So why would we be doing stuff with fiber, right? Like why would we be absorbing it, right?
Yeah.
I mean, I can see how that can make you think it'll be slower because we're turning the fiber into other stuff. But yeah, we're not absorbing, or that's not what the bacteria is doing. It's not causing absorption. And the fiber we did not talk about fiber itself getting absorbed. And then slower, I could see how that can be something that could kind of make sense, but generally speaking, the rate of reactions would only be affected by like the presence of a catalyst, kind of like the kinetics kind of thing.
Mm-hmm.
So there you go. 3 reasons for B not being right. So
I'm
what?
very passionate about B not being an answer.
Yeah. Because yeah, the more reason you have, yeah, it doesn't stand up to the standards. Yeah. So okay.
Okay. Um, then immune tolerance, I don't think came up anywhere.
Maybe
Um, I don't know. Question mark on that one. Increased amounts of propionate.
so so yeah, I wrote it pretty sloppy on the bottom. I apologize for that, but I did kind of there is something that they kind of mentioned with
That fiber
somewhat related
bacteria.
to, yeah, keep going with
Um,
that part.
like, okay, so inversely proportional to inflammation.
Hmm.
So the more of this than there is, the less of this. But also just from looking at the passage, you can see the propionate it isn't on the table anywhere that it's like CD.
Mm-hmm.
So C's wrong.
Mm-hmm.
B's wrong. Increased polypeptide digestion. Maybe.
So
A.
I mean you're breaking down um protein, right?
Mm-hmm.
Um, did we mention anything about that in passage?
In which one? In breaking down protein? I don't think so.
Yeah. So but we did say something sort of related to immune.
Healthy individuals versus host immune response to GI tract bacteria. So decreased immune tolerance actually does.
Uh-huh.
So CD affected individuals. Oh, it doesn't. Wait. Okay, potentially. So D?
Yeah, right. Because the main difference between CD and people without CD is people with CD have that like autoimmune kind of reaction. But yeah, you're gonna get D.
Okay.
And it was the inflammatory. That's the reason why. So that's
And
why I would write it out like how I wrote it out there. Um, I mean, I would do it more neat, but just
because that like helps you because once you move on from reading it, these things kind of like slip your mind, but once you have it written
it's
out,
like
it
8,000 different, acronyms. Like this one isn't even like that bad, but you're gonna see some with like a trillion different acronyms. Like so it really helps. But but yeah. And you can see like how the nuances in language play a major role here.
okay. Right, and B was the answer that I picked, so it's like critical thinking of it. Like I guess I'm wondering is you had an issue with B right away because of absorption.
Um, I mean, it's not like I particularly had it out for B. It's just because you mentioned that you liked B or something like that. That's
Mhm.
when I think I mentioned, actually it's not that good. But are you saying like so yeah, you're saying that I didn't like it because of absorption?
Well, I guess I'm just saying like when we're really taking the time to like chew it down, like I can see why like we went through each answer choice and we saw which one was right, which one was wrong. But like if it's on an exam and I'm doing it quickly, I guess I don't know. I still think that like I saw dietary fiber in the passage, but then again, this one was there too, the immune response. So
That's like the most common thing that people do. They try to choose an answer choice that's verbatim in the passage, and that's like 99% of the time going to get you the wrong answer. It'll get you the trap answer. It'll get you the answer because to make these questions, you can't just make it so like A or like 90% choose A, right? You want it to be a good breakdown, like 60-40, something like that. So how do you make a good trap question, right? You include a word in it that's directly mentioned in the passage. And that's how most people handle these types of passages or passages in general. Like they just play mix and match, but you want to be able like that's why I also want you to like put things in your own words
mhm.
so you're not going to be as susceptible for when seeing that they're using the same words. But yeah, I have three reasons why B isn't good. They're asking what do the like they're essentially asking us what does a bacteria do? And we know from the passage that the bacteria takes fiber and breaks it down into butyrate, these three things, right? The three molecules. And that the three molecules here. So what the bacteria does is they digest fibers, convert them into these three things, which uh, modulate the immune system by attenuating, which means weakening
Mhm.
the inflammatory response, right? So right away, I know that, okay, we that's what we're doing, right? So first of all, that's what I'm going to be looking for. And something I could try with you is because this is something that I also like to do with students, like I block out the choices so you are able to come up with your own answer. Because if you do that, then you're less susceptible for the wrong ones. But here I'm seeing that fiber is broken down into these three like things, and that's what the bacteria does. So dietary fiber, sure, we mentioned that, but we said digestion, not absorption. So
Mhm.
that's out. And slower, like nothing's really indicating to us any change in rate. It could be it's a good trap answer choice because if we're saying that fiber is broken down into like this stuff by the bacteria, and if you don't have the bacteria or something like that, right, your microbiome is messed up if you have CD, then it wouldn't change like the rate at which it's happening, right? So those are the three reasons: absorption and the fiber being absorbed, and uh the rate, the speed of the reaction. Um so, and yeah, it's mostly here.
Yeah. Okay. I think that was it for this passage. And then this one was like, I know the structure of a neuron, I know like functions of it. So I think I was between A and B here.
Okay, so
Uh,
let's rephrase the question. Like, let's put the question in your own words, so we, I guess it's super important to make sure that we know what we're being asked.
so in contrast, should I just read it out loud first and then try to like see where my mind takes me with this one? Okay. In contrast to messengers in classical endocrine signaling systems, chemical messengers in paracrine and autocrine signaling systems are. So it's saying like in like basically what chemical like some quality of chemical messengers in endocrine versus paracrine and autocrine. Like what can we say about them?
Mm-hmm.
Um so yeah, so it's just like asking about the characteristics.
Okay.
Um
So what
I
can,
guess like we can see maybe what values we can associate with like endocrine, paracrine, and autocrine and see if it like lets us get to an answer like we can process of eliminate.
yeah, so let's think about what is endocrine.
So see, like I know, but I don't know how to define it. That's like I feel like that's my issue a lot with the MCAT is that I understand the topics. I know like
Yeah.
autocrine and paracrine, it's like different like parts of like the nervous system and like the response of like function is like if something gets like stimulated, it's paracrine or
So
autocrine.
yeah, so I know I'm asking very like, open-ended, vague questions, but I don't expect you to like give me the right answer necessarily. I want to just have you like think out loud so I get an idea
I
of,
know, but I'm saying that like if there was a question where I think this is like a lack of information, like just knowledge, I think this might be it where I need to review like endocrine, paracrine, and autocrine and see like I don't know, but maybe if you could like quickly summarize them for me, that would be really useful because if I have the like information, I can probably
yeah. So
answer the question.
so yeah, so before that, I'm just gonna ask like, what would you say these things mean?
Um, signaling. Oh well, that says that.
What's
Um,
endo mean?
endo is like internal, like within. It's like endothermic is something that comes to mind.
And then we have para and auto.
Um, auto is like something is acting like on itself. So if it's like a cell or something,
Okay. And para, if you don't know, it's fine.
it's like nearby.
Okay, you do know. Okay, good. Okay, so there's something that the endocrine does. That,
Endo, like I'm like within, so I don't know how specific is it. Is it like within the body or like within a cell? But that's what we said autocrine was.
oh yeah,
Is endocrine like on a bigger picture?
so let's see, right? Um, okay, so paracrine nearby, auto self. So endocrine must be not affecting itself. and not what?
Nearby.
Yeah.
So something distant.
Yeah.
Hmm.
And if it's something distant,
So
yeah.
if it's something distant, then it would be transported by ducts, right? Because it would go through the ducts to the receptor.
Let's say you're in a let's say you're in a human body somehow. You're shrunk yourself and you're in a human body and you wanted to go from one organ to the next. And what would be the best subway system to use for that, to travel like that?
From cell to cell or like throughout the body.
Throughout the body.
Vessels.
Yeah, the blood, right?
Mm.
Because so you see how like you were thinking like, okay, I don't know this, like I should like read up on it and stuff. And that's fine. You could totally do that. But I want you to just see how you don't need to know everything. You can still logic it out and make the proper inferences, right? So
Mhm.
so you knew that endocrine meant internal. You knew that paracrine is nearby, and you knew that autocrine is self, right? And if they're asking us what's the difference between them, right? That must mean that the endocrine is not nearby and is not affecting itself.
Okay.
And so how do you
I
trans
don't
yeah,
really.
yeah.
Are you saying that if they were close by, it wouldn't go through the blood vessels? Like it would just travel to the other, but how would it have reached the other cell?
It
That's
so like if it's nearby, it can just release it into like the interstitial space. That could be one of the things that it can do. But
Mhm.
the yeah, uh that could be something that happens.
Okay.
Um
So if it's not, then those two wouldn't be transported by blood vessels.
yeah. Um
Not like, okay, so that's why like blood vessels make sense. The other ones, I don't know. I guess ducts I was like confused about. Like I kind of didn't really, right.
ducts would be exocrine.
Uh-huh. And then I feel like only is usually like a trick word from what you've said and what I've seen. Is that like why is it only neurons that secrete it?
Yeah, yeah.
Uh,
Like only is usually uh like sus. So
mhm.
um but yeah, so yeah, on
And
the
then I feel like they would all bind to cell receptors.
um yeah,
Or I guess the paracrine and autocrine for sure would, because if they're on like a cell to cell or like within the cell, like they have to bind to certain receptors. So the fact that it's not likely is kind of like I'm not sure if they do for endocrine, but it for sure has to for like paracrine and autocrine.
uh-huh.
So I guess yeah,
Yep, yep. So
like I wouldn't have known C is exocrine, but I can see why A and B are wrong now.
yeah, yeah, yeah. I just told you that like for you to know for next time and stuff, but
Okay.
yeah, I just wanted you to see that we can still figure stuff out. So like, I always stress tolerance of uncertainty. If you don't know the answer, it's okay.
Mhm.
That's the best place to start. Um, and and also not having your answers in front of you gives you another chance to think it through,
Yeah.
But you saw how we just like logic that out. So that's what you want to do. Um,
What did
I
I?
don't want you like to be thinking, I need to read up on this more because that can lead to like a fallacy where people just like constantly are like reading content and like feel like they're never done with content and there's always a way.
I feel like this might be a
Oh, what's this?
trick answer just because like when you think of like signaling systems, your mind automatically goes to like neurons and synapses. So then it's knowing like the only is kind of a red flag.
Oh, okay. I mean, when they say endocrine and you think of the endocrine system, that's where you have like hormones traveling through the blood, right?
Hmm.
Um, and exocrine, like the example would be like the hepatic portal, right? But since you're saying, yeah, uh, endocrine and then paracrine and autocrine, like they are not really mentioning anything about the neuron stuff.
Okay. Which membrane transporters electrogenic and translocates a net charge? Um, okay, so I know I picked NaCl for this one, but I don't think that that necessarily like generates a charge because that's like what's supposed to be happening.
Mhm.
And then something like A, for example, where there's positives on both sides of it, then the overall thing generates a positive charge. But I don't know, then translocates a net charge across the membrane. Like, wouldn't NaCl have a net charge because like there has to be like an equal amount on both sides of the membrane?
Okay, so we have this sodium plus H plus exchanger, which I guess means that it would, like, let's say, pump sodium out and pump protons in, or vice versa.
Right.
For the word from the word exchanger, we have the co-transporter. What is that? How is that different from exchanger?
That it's not like a direct system, like there's something else there that's allowing this transport. Or no, it's transporting both of them. But how is that different from exchanger?
Um, think about the difference between exchanging something and doing it with like CO.
Co is like something else is there that's
Uh-huh.
facilitating.
So it would be like so like, okay, exchanger, we're exchanging sodium for protons.
Right.
Co-transporter, we're not exchanging them, so they're going to go in the same direction. Like sodium, that was horrible.
Oh, so I forgot. The thing is NaCl is that like the sizes change, so it's easier for one to get through than the other.
Yeah, so a co-transporter means that they're going to go in the same direction,
Okay.
right? Um, and they so what do they mean when they say electrogenic?
But it has a charge.
Yeah, that it produces some type of electric field or electric, uh, pretty much that. And then translocates a net charge across the membrane. So so yeah, go from there and tell me your thoughts.
Translocates a net charge. So then would an exchanger be better than a co-transporter because it would have a net charge across?
Mm-hmm.
It's like for every sodium that gets taken out, a hydrogen comes in, or vice versa.
Mm-hmm. Yeah. So
Probably means that there has to be some kind of equilibrium, and that's like a part of net charge.
yeah, the net charge of this would be 0, right?
Yeah. Well,
Yeah,
does the fact that they're both positive not make a difference? Because
they do. Because you're removing a positive and you're adding a positive.
adding a positive, okay.
And then here, right, you're adding a positive and you're adding a negative. Your net's 0, right?
So these are both wrong.
Correct.
Translocates a negative charge, a net charge.
Okay.
Is A also wrong because it's not a transporter? Like inherently, it's an exchanger.
Um,
Or does that not?
when you say inherently, are you talking about the exchanger part or the NaH plus part?
Well, now I'm looking at just the wording,
Mhm.
how it says exchanger, not transporter, and we just like spoke about how there's a difference.
Yeah, an exchanger means that they're gonna go in the opposite direction. It's as if you're exchanging sodium for you give away sodium, you get a proton H plus.
But it's still a transporter.
Um,
Like
well,
by like
uh,
I get what you're like saying specifically, but would it still fall under the umbrella or no?
I guess it would be for like a transporter, but
So no, okay, so like what I'm getting at is that like because it says exchanger, could I have immediately crossed it out or no?
oh no.
Okay. All right.
Because what if it was an exchanger with NaCl? Or what about the one that's in neurons, right? The potassium sodium one, right? Like 3 sodium ions come out, 2
Do come in.
potassium comes in. That's an exchanger, right? So exchanger just means that uh something is coming in and something else is coming out.
Okay.
Co-transporter, they're both either coming in or both coming out. And I guess transporter, just the word itself, is not going to involve more than one thing. So
Right.
D, glut2,
So
facilitate.
the fact that it's only one thing coming in or out, regardless of like the charge of glucose, it would translocate a net charge because it's like outside of the cell of the membrane and it's coming in or from in to out. So it would change like the overall charge somewhere.
Well, well, glucose wouldn't have a charge, right?
But with the glue too.
Um, so the glut2 is just some type of transporter that transports glucose, but glucose has no net charge, right?
So it wouldn't change anything. So how would so then if that's true, then C, because then you still have the sodium
Yep. C,
that
you
charge.
have sodium positively charged coming in or out. Glucose.
Glucose going in or out, but that one doesn't have a charge, so the overall charge is now positive. Huh.
Yep.
Okay, so I need to know that glucose doesn't have a charge.
Well, yeah.
That's just the takeaway from this question is that like now I know what like exchanger, transporter, co-transporter, like what those specific ones mean.
I mean, those are, I mean, glucose not having a charge, you should know.
It makes sense. Like I know the chemical like structure of it.
Yep. Yep.
So
Yeah.
that's just okay.
Because yeah, that's not like what I would say the takeaway is. The takeaway is an exchanger means that they're going to go in opposite directions. A co-transporter, think of the word co, like a cooperative, etc.,
Yeah, like that there's
going in
two
the same direction.
happening.
Yeah, are going in the same direction. And transporter is just one thing that can either
So
come.
okay. Yeah.
So, but yeah, glucose, you should know, doesn't have a charge. In fact, that's what the first step of glycolysis does. It imparts a charge, making
Uh,
it can't really, go back out through the GLUT2 or GLUT4, I think.
sounds good.
Yeah. But again, you see how we're
Oh.
using like the nuances in language to answer
Hmm.
this. We're not using our own, whatever knowledge.
Previous answer was B.
Hmm.
I think I like I don't know, I
What
saw
what
that
word
I
can
knew that NaCl like doesn't have a charge.
So what word can we change in B to make it right?
Exchanger.
Good.
Lack a nucleus. Well, I knew neurons weren't circular, so that's wrong. Um, Blastocytes, like I was thinking like bacteria if I like remembered anything about A or B.
What do you imagine so a shape is like a flat disc with a concave center. What does that mean, concave center?
So like goes in.
Huh?
We spoke about that in physics last time. I think like we were talking about convex and concave.
Yeah, and it all comes back to, etymology is also really cool because like cave, cavity, right? They're like holes.
Yeah.
So
I mean, then I was thinking like mono would probably have like a singular like function and maybe lacks certain organelles
what
like
what what type looks like that flat disc with a concave center? Like what if you picture it, what do you picture?
a flat disk with a concave center, like lacking a nucleus would make it have the concave center, I guess.
I don't know if that is what makes
Okay,
it.
well, all right, I don't know. I like nothing from the three of these comes to mind, but
Oh,
I just feel like the
yeah.
I don't know, the flat disk of it all makes me think that it's C. That's where my mind went.
So I kind of drew it here. Maybe that can help.
Isn't A the one that has like the like body and then the tail? I like genuinely don't know.
So I changed the color.
Oh, a red blood cell?
Because that's what they look like, right? Have you seen a picture? Like,
Yeah, yeah, yeah. I always think that they look like little like lifesaver gummies.
yeah, yeah. So, that's the whatever the biconcave thing. And here I'm putting an image of it in the chat. But yeah, they don't have a nucleus, they have a plasma membrane. So
Mhm.
and they lack most organelles, and then the flat disc with a concave center, that's going to be your red blood cell. So which one of these choices is a red blood cell? Or which ones aren't?
Like fibroblasts and erythrocytes both have multiple functions.
Oh, I thought you're.
I mean, yeah, I am getting there that they're like singular. Like, I don't know. I still think that like everything leads me back to C. I don't remember which one is associated.
So your mom's a dermatologist, I'm guessing.
Mm-hmm.
So what is it called if you have your skin turns red?
Irritation.
Anything with a E?
Oh, erythema.
Yep.
So,
So
yeah, that's like a rush of red blood cells going to the area.
yeah, so arith just is. Did it tell you about how like 60 to 80% of words in the English language come from Greek Latin roots?
I mean, you didn't tell me, but that does make sense.
Yeah. So that's why a lot of times you can figure stuff out without knowing the word. So here, erythema is from Greek erythros, which means red. And our answer we're looking for is red blood cell, right? So it must be erythrocytes. We know it's not neurons.
And-
We know monocytes are white blood cells, right?
Oh, that's why.
Hmm.
Okay.
Yeah.
All right.
So everything that I'm showing you here is just like, it's not like to be like, this is what you have to know, this is what you have to know. I want you to see like how we deal with not knowing and then kind of figuring it out.
Okay. That makes sense.
Yep.
Right.
But yeah, pretty much I wrote like an SAT guide a long time ago where I talk about learning words, but yeah, it's either 60 or 80%, but they're all from Greek and Latin roots. So that means if you know like 14, 15 Greek Latin roots, you can figure out most words. Electrogenic, gen, Gen, like Genesis, the beginning, like, in the Bible or whatever. So that's
Yeah.
the beginning. So electrogenic starts that electric. Yeah. So, yep,
Okay.
there you go.
Makes sense.
Mhm.
See, that answer is like two.
What is that? Have all the character. Oh, yeah, yeah, yeah.
Okay.
Oh, yeah, this is erythropoietin, which is something that.
Also red.
What's up?
Also, well, they're saying red bone marrow. So I
Yeah,
remember I saw this question, and the next passage is literally about erythrocytes.
yeah, yeah, yeah.
That didn't raise any red flags because, okay.
Red flag, and you even said red, so there's a lot of red going on. I'm wearing red. My glasses are red. Today's a red day. So, in the
Okay.
stock market, it's a red day today.
My phone case is red.
There's a lot, yeah, there's a lot of that happening right now. So, okay,
Okay.
so yeah,
Okay.
let's,
Um, I honestly I'm not sure how many I got wrong. Oh, so it was only one question for this.
yeah.
I
So,
don't know, is it really worth it? Because I was missing like the part where
yeah, let's see like another one.
um yeah, so
For
these are
that.
all
So there's two for this, and let's see like how many after. Like, I'll count. So, okay, one. That was one for that.
should I keep going? These ones are okay.
One for that.
One
One.
and
One for
one,
that. Okay.
two,
For this. There's probably like one more.
one, two,
Only two for that. Okay.
one, two, three.
Three for this. Okay. We honestly could do any of these. We don't need to do.
I don't know. I feel like here it was closer to the end. I might have like checked out a little bit, but
Yeah, so that would be good to do your blind review on. But yeah, we can do one of the other ones.
do like this one. Oh, was this only I only had one? I guess let's start with the first one. It was this one. So I think we only have time to do these ones and then
Mm-hmm.
okay. So let me read it out. Um, Selective serotonin reuptake inhibitors, SSRIs, are drugs that alleviate symptoms of depression by blocking the reuptake of serotonin from the synaptic cleft, thereby increasing the amount of time that 5-HT serotonin remains active. Okay. Elevated levels of 5-HT within the synapse are associated with feelings of well-being. Conversely, low levels are associated with depressive symptoms. That makes sense. So more serotonin equals happy, less serotonin equals sad. That's like, I don't know, that's like a known, like out of all the,
Um,
of all of like the hormones, I don't know, that's one of the ones that like structurally people understand.
so 5-HT is the SSRI that blocks the reuptake of serotonin. Any proportionality we see here?
Well, no, I don't think that that's the SSRI because that's like the R4 one.
Wait,
The
sorry.
5-HT is just serotonin.
Oh, okay, okay. JK, okay. So, oh yeah, okay. So it is the serotonin. Okay. Um, what's the? Oh, okay, okay.
The R4 protein.
Okay. Yeah. Okay. So sorry. Yeah. Okay. I guess. Yeah. Okay. Continue. Sorry.
Recent studies have shown that SSRIs can also mediate their antidepressant effects by increasing brain levels of certain cytokines, including the interferon gamma IFN, which directly induces the expression of proton P11 in neighboring neurons, then interacts with 5-HTR4, a serotonin transmembrane receptor.
All right. So there's a lot of these acronyms here. So let's.
Okay, I found this on the web for turn
I was like for a second, like, hey, your voice completely changed.
Every time I say something, even remotely, it happens all the time on this
Yeah.
computer. I don't know.
So, oh yeah, so okay, any proportionality we can say here.
Okay.
Or maybe I'll say,
So okay, we can see that we have SSRI with okay, increasing brain levels of interferon. Okay. Um, induces so then IFNY with the P11 and that interacts with the receptor.
so let me think about how to put interacts with.
Well, because I don't think it tells us until we look at the table what the relationship is. Like we don't know if it's a positive or negative correlation.
So okay, let's see what we get. So good job, good job. So yeah, figure 1, 5-HTR4, protein expression in plasma membrane enriched fraction, MEM, of hippocampal lysate and total hippocampal lysate, TOT, from P11 wild type WT or P11 knockout KO. So yeah, this is when the BB passages get to be ridiculous because there's so much going on here. But that's okay, we could still do it. So let's see. Yeah, so why don't you evaluate figure 1?
So expression of lysate of membrane enriched fraction versus total lysate from the wild type versus knockout. So knockout means it doesn't have any P11, right? Or it's all P11. No, knockout is get rid of. And then wild type is we're not sure
wild
how
type,
much it's expressed.
yeah, wild type would be the normal amount.
Okay. All right. So they were like when they do a study like this, they're using it as like the like dependent variable, I guess, or like if it's not there, this is what the results look like.
So the
It's
see the
normal.
y-axis is doing this as a percent of wild type. So this is like they normalized the wild type.
Okay.
Like whatever it is for the wild type, they normalized it and made it like 1 or 100% here. And everything else is compared to that.
Okay.
So
I guess like there's like no change between the total and then for the membrane enriched, the knockouts like 20% lower.
mhm. So
I don't know, that's like really the only difference I can notice is that like maybe there's a correlation between or negative correlation between knockout
mhm,
and mem.
mhm.
That's I don't know, really the only
So we could say like inversely proportional to
mem. Okay.
5-HTR4 in mem, maybe something like that.
Okay. And then effective serotonin on cyclic AMP levels in cells transfected with the 5-HTR4 or P11. So camp levels are the same or like none for control. P11 on its own doesn't really do anything. 5-HTR4 raises it to 40%, but it gets much higher when you combine both of them.
Okay, so now the camp levels is related to the percent change from control and P11. We have a slight bit. When we have the 5-HTR4, it goes up. We have more camp. being expressed, and then with the P11 you have even more. So I would maybe write like oh damn it. Okay, so I'd write like oh god, I keep doing this. Sorry. Because when I annotate, it's going to like the wrong tool. Okay, this should work. Okay, so 5HTR4 proportional to camp levels and like P11 also produces a high amount of the camp. So okay, cool.
Like a very like
Mm-hmm. Oh Jesus. There's a lot. Okay, let's do it.
studies have shown that analgesics such as ibuprofen inhibit the behavioral antidepressant responses normally observed in mice undergoing chronic SSRI in administration. So ibuprofen is like the IFNY, no? Because wasn't that one something that also decreased antidepressant responses? Oh, wait, I read that somewhere else. Induces the increasing brain levels, immediate their antidepressant responses. So then we can say that there's some positive relationship between IBU and IFNY.
So we know that IBU, from what they're telling us, gives us or inhibit the antidepressant effects.
But isn't that also what they were saying about the other ones?
When you say that, because ibuprofen is just like,
I know what it is. It's like taking away like inflammation and stuff, but look at like can mediate their antidepressant effects, including interferon gamma, IFNY. So inhibit the behavioral antidepressant responses. So there seems to be some sort of relationship.
Yeah. So yeah, what I see so far is that it inhibits that antidepressant effect on mice that have been on SSRIs. And then I think afterwards they'll tell us more about the relationship with IFN and stuff like that.
Okay. Inhibit the behavioral response observed in mice undergoing chronic SSRI administration. Figure 3 shows the effect of citalopram on an SSRI and IBU on IFNY expression in mice.
Yep.
Figure 4 shows the effect of IBU on P11 expression in the presence and absence of treatment with CIT. In both figures, VAH represents the group given the control solution. Okay. So, do we know what citalopram is?
They say that it's just an SSRI.
Oh, and I thought it was like saying like the effect of this, and that one. I couldn't tell that they were saying that it is an SSRI. Okay. So the SSRI on its own obviously has really high levels of antidepressant, right? Of brain levels of certain.
Yep. So we could say.
Wait, but mediate their antidepressant effects by increasing this. So SSRIs on their own increase this a lot. With
So the CIT, oops.
Oh, so that makes sense because CIT would mean that it would need a lot of this.
Yeah, so CRT.
But then something like the ibuprofen with CIT, since it inhibits the antidepressant response, then it doesn't need it. So I was wrong, and there's actually negative correlation between IFNY and the ibuprofen.
Yeah. So we see that there's a positive with the IFN for the citalopram, the SSRI, and the ibuprofen, as they, told us, we see that it's having that negative effect. So I can add on to this over here, 1 over IFN gamma.
Uh-huh.
Yep. So,
All right. And then now we have to do the fourth.
yep.
The effect of ibuprofen on P11 expression in the brain and presence or absence of citalopram treatment.
Okay.
So we said VH was the control. Right? So that one has nothing.
Yeah, let me see again what that was with that VE. Okay.
Control solution.
They don't even tell us what it stands for. Okay, whatever. So cool. Control. All right. And so we see in the control, the the black bars are the control, the sit is the ones given the SSRI citalopram, and we see that in, so maybe like, let's see,
I feel like with ibuprofen, everything's lower.
citalopram would be proportional to P11 expression in this control, and ibuprofen inversely with the P11 expression. So there's a lot going on here, but what we can see is that the more SSRI that you take, the higher amount of IFN gamma, which means a higher amount of P11, which means less of this 5-HTR4 in the MEM, and that the 5-HTR4 also means more camp. P11 means more camp, and ibuprofen means less of the antidepressant effects. So essentially, you should see the inverse for all the things that we said the SSRI does. So it's inversely proportional to IFN, where and then we have sit, the citalopram.
Can I ask a question? Like, if you were to actually take a test, would you walk through and make all these connections? I feel like it's very tedious and you don't know, like, especially if you're saying only spend like 10 to 15 seconds the first time reading each graph. Like,
Yeah. So when you're first reading it, you're gonna try
so you
to.
don't know if this is going to come up in any of the questions. Like, if we're looking at the question, it says, what does like 5-HTR4 do?
Well, it's still going to. Okay, so yeah, I
Like,
would.
it's definitely going to help. I'm not going to like negate it. I'm just, I guess, on a time crunch, would it really be necessary?
I would say yeah, because there's different schools of thought on this. What I would do is like the same thing, but with the 15 seconds for the graphs and the passages. So if they ask you a question on it, then go back to it. But as I read a passage, I definitely write out these proportionalities, because that means that if I need to go back to something, I don't need to go back to the passage. I can just go back to what I've already written down. And that's like,
Saves you time later on. I understand.
yeah, exactly. Whereas a different school of thought is like, what if I read the questions first? Now, sure, that might be a possibility, but if you read the questions first, you're going to have to go back and look for where in the passage it is that it's relevant. And if you haven't read the passage, you're going to spend that time reading the passage and going back to the question anyway. So I think that it's better
That makes sense.
to do it like this. And yeah, and when I write out proportionalities as well, because that way I don't need to, have to analyze all these different acronyms, right? Because here we have 5-HTR4, we have P11, we have IBU, we have CAMP, we have IFN gamma, we have P11. Like, there's so many things going on here, but writing out the proportionalities means that I know if something goes up, if the other thing goes up or down, etc. So yeah, I definitely do write it out like this.
All right. And then for this answer, I like from this,
Mhm.
or no, from this, like I know it has something to do with proteins
Uh-huh.
and protein P11 interacts with 5-HTR4. So then I'm gonna say it's something to do with like either A or D because of that. And then this is like from the back of my mind something with like camp that we learned in school that it has something to do with G proteins.
Okay.
Makes me lean towards D, but I'm honestly like unsure.
So tell me again what led you to eliminating B and C, like you're thinking either A or D.
Well, there's a lot about proteins.
Okay.
So,
So there's one word in that in that sentence that uh will help.
well, it's a transmembrane receptor. So
Transmembrane.
then by the same thing, like you can say that it's a protein, so that gets A and D, but then receptor gets you B and D, but since B isn't a protein, it's D.
Well, yeah, because you want so the G protein coupled receptors are going like there's a lot of stuff in the transmembrane of it, right? Whereas for the other ones, they're just receptors on the outside.
Okay, that
So
makes sense.
the
Yeah.
transmembrane receptor is what gives it away.
My previous answer was A. So see, I knew it was something to do with the protein, but I didn't rule out the kinase.
Yeah, the kinase is a very high yield thing, so you should know about STY.
Catalyze the addition of a phosphate group to protein. Okay.
Yeah, it could also remove it.
Okay.
I'll tell you more about it like next time, but it's pretty high yield.
Okay. Sounds good. I think this is the last one for this one, so we can do it and then wrap up.
Yep.
Expressions of P11. Oh, what comparison best determines whether IF and Y is necessary for antidepressant induced increases in the expression of P11? Induced increases. What does this one say?
Oh, citalopram proportional to P11 expression in VEH.
Okay. And then okay. It was based on P11, it was this one.
Okay.
Or which comparison best determines whether it is necessary? So I guess they're saying like which one would you look at to be able to deduce it?
So yeah, it's asking, expression levels in expression levels of P11 in what would best help us figure out whether INF gamma is necessary for the increase in expression of P11 or not necessary.
Mm-hmm.
And it's antidepressant induced.
So they would. I feel like I don't know, these two, they're all treated with SSRIs. That's kind of the point of the experiment. So antidepressant induced increases on the expression. So they would all be tested by SSRIs. I don't think that's necessarily specific enough. Oh, so if it's necessary, both treated with P11 if the IF and Y is present or not? A.
Well, is
Is
P11,
that too straightforward? It's being expressed, so it can't be treated with it because that has to be the outcome, is the level of P11.
yeah,
right?
so it can't be treated with P11, right?
That's why A is wrong.
Mhm.
Both treated with an. In the graph that was showing P11, it was looking at the SSRI
Mhm.
versus ibuprofen. That's for IF and Y. Sit versus P11. It's not that they were being treated with this though.
Mhm.
So, like, if there was an answer choice that was saying like ibuprofen versus SSRI, that would make sense to me.
Ibuprofen versus SSRI.
From the P11 expression because that's what it was saying, expression percent of control. So we have the control group
Okay.
was on SSRI.
So
Maybe this one. While I feel like that could potentially be wild type treated with IF. Why? What's the difference between? Oh no, because it's no, this makes sense because this is like the control, the dependent variable, well, no, the independent variable is whether Fi and Y is present.
yeah,
Um,
yep.
which makes sense for this. This one, it's saying that it has, one of them has it, one of them only has SSRI. I don't know if like you can necessarily draw the connection there. So I really like that this one, it's both like just, okay, yeah, B. This one, it's IF and Y is absent. That's the
Yeah.
QD.
Because we're being asked, how do we know that the SSRI produces an increase in or sorry, how do we know that an SSRI that leads to IFN causing an increase in P11,
Okay.
right? So that means that we can't well, we need to treat both groups with the SSRI because we're being asked like
This one was really tricky. I feel like it's like a reading comprehension thing almost. And like
a lot of
your
this stuff
mind immediately goes to A because it's pretty much restating the question,
Yep.
like the way that it's asked. But then that's what you were saying before, that it's like too simple. And
Yeah.
then you start looking at it like treated with isn't the expression.
Mhm. Yeah. So yeah, you want to see that the question is, so if you can just hide it for a sec, hide the solution. So this SSRI, we want to see if this SSRI turning into this P or increasing the expression of P11, if that if we can see that IFN gamma plays a role in that,
Okay. Yeah.
right? So because we know that an SSRI causes more P11, but what we're trying to find out is if IFN gamma is necessary for that P11 to go up, right? So we need it to be treated with we need both groups to be treated with the SSRI, and we need one group to not have IFN and one
Okay.
group to have IFN. That
Yeah.
way we can see if the SSRI increases P11 with IFN or without it.
Okay. That makes sense.
Yep.
That one was tricky, but like once you like started really carefully looking at them, it made sense.
Yeah. So yeah, you've seen from today just how much like the word
Reading
is
through.
nuanced and subtle, like how subtle it is. And and about how a lot of those questions, like, okay, so when you feel like you see something and you want to like kind of give up or whatever, like you know, you feel like you haven't learned it or you haven't learned it to the extent that you want, I would say just to stick around and try to be okay with that uncertainty and try to logic it out. And I think that that's going to be, you know, to your advantage because like theoretically, if you took the MCAT open book, like it still won't mean that you're gonna necessarily score super high, right? Because
Mhm.
it's still mainly a critical thinking test. But yeah, you did a good job. So for reviewing that, for the blind review,
Just continue the way that we were doing it and reasoning through each question.
yeah.
When can you meet again? Like,
Mhm.
I guess I'm gonna spend tomorrow blind reviewing it now that I understand how to do it, and then I'm hoping to take another one on Saturday. I feel like because that's the day of my exam, I want to get into the habit of like every Saturday having one.
Mhm.
Um, yeah.
All right. So
So potentially, I don't know, I can meet, yeah, maybe Friday we meet.
um this Friday you're talking about?
Yeah,
Let me see.
because you said you wanted to start doing like two
Yeah,
times a week,
yeah.
which I completely agree with.
Yeah, let me okay, let me see something. Um okay, so I'm going to
You send me the Calendly, I can take a look at it. I was insanely busy and didn't get to it,
yeah.
but
No, so I can
I'll,
put you in for Friday. I'm just gonna need to move another person over. But
okay,
let's have let's do the 6 p.m. Eastern work.
okay, sure.
Okay.
Sounds good. All right. So see you on Friday.
Uh yes. All right. Take care and yeah.
Okay. All right. Sounds good. Bye bye. I'm gonna add it to my calendar now.
Yep. All right, take care.